diversity of animal tissue. Yes, the first one is squamous epithelial tissue. Yes, see, squamous epithelial tissue, they form a covering of the body, and the cells are thin and flat and single layered, arranged on the basement membrane. Yes, in the right side we can observe the cells are thin and flat and single layer arranged on basement membrane, and presence of central nucleus surrounded by cytoplasm. There is no space between two cells. These are all about squamous epithelial tissues. Yes, they form the covering of the body and presence of cells. The cells are thin, flat and single layered arranged on the basement membrane, and presence of nucleus surrounded by cytoplasm without intercellular space. Yes. The next one is connective tissue. Dear students, we know about connective tissue. The blood is a fluid connective tissue. Blood is a fluid connective tissue. It contains plasma and corpuscles. So you know about plasma. Plasma is a light yellow colored fluid which stores water, salts and some enzymes. Yes, and the cellular portion of blood containing RBC and WBC, red blood cells and white blood cells. The red blood cells also called erythrocytes. Yes, these erythrocytes are oval in shape. Yes, in the right side we can observe the red blood cells. So these red blood cells are oval in shape and numerous, which means great in number, and numerous, and enucleated, which means absence of nucleus. Yes, RBC is red blood cells. These are all oval in shape and it's also called erythrocytes, and they are numerous in blood and enucleated, which means absence of nucleus. The next one is WBC, white blood cells. Yes, these white blood cells also called leukocytes. These leukocytes or white blood cells are two types. One is granulocytes and agranulocytes. See, the granulocytes include neutrophils and eosinophils and basophils. Yes, in right side we can observe the neutrophil is a granulocyte. It's showing multi-lobed nucleus. And the eosinophil shows bilobed nucleus. Bilobed nucleus. Yes, in the right side we can observe both neutrophil and eosinophil. The neutrophil showing multilobed nucleus and the eosinophil showing bilobed nucleus. And the basophil cell shows S-shaped nucleus. S-shaped nucleus. Yes, these are all about granulocytes. Yes, these granulocytes are the human cells which kill the pathogens, and these are all types. Neutrophil, it shows multilobed nucleus, and eosinophil which shows bilobed nucleus, and basophil which shows S-shaped nucleus. These are all about granulocytes. The next one is agranulocytes. These agranulocytes include monocyte and lymphocyte. Yes, monocyte and lymphocyte. Yes, see, we can observe in the right side the agranulocyte, the monocyte showing kidney-shaped nucleus. Kidney-shaped nucleus. Kidney-shaped nucleus. And the lymphocyte shows spherical-shaped nucleus. Spherical-shaped nucleus. Yes, this is all about connective tissues. Yes, blood is a fluid connective tissues containing plasma and corpuscles, and the cellular portion of the blood is RBC and WBC. RBC is red blood cells, is also called erythrocytes. So these are all oval and numerous and enucleated. And WBC, white blood cells, also called leukocytes. These are all two types, granulocytes and agranulocytes. The granulocytes include neutrophil, eosinophil and basophil. The neutrophil shows multilobed nucleus and the eosinophil shows a bilobed nucleus, and the basophil shows S-shaped nucleus. And in agranulocytes, it includes monocytes and lymphocytes. The monocytes shows a kidney-shaped nucleus and the lymphocyte shows spherical-shaped nucleus. These are all about connective tissue. The next one is muscular tissues. Yes, the muscular tissues include skeletal muscles, smooth muscles, and cardiac muscles. The cells are cylindrical. And unbranched. And these are all voluntary muscles, which means the muscles coordinated under our control. Voluntary muscles. Yes, and sacrolemma. See, sacrolemma, which means structure of muscle cells, that is called sacrolemma. With thin sacroplasma, which means cytoplasm of muscle cells, that is called sacroplasma. Showing multinucleated, yes, we can observe, containing multinucleated peripheral nuclei. And presence of numerous, which means great in number, numerous myofibrils, which means muscle fibrils. It shows alternate dark and light bands. These are all about skeletal muscles. Skeletal muscles are cylindrical and unbranched, and these are all voluntary muscles. and sacrolemma with a thin sacroplasma, and it shows multinucleated peripheral nuclei, and numerous myofibrils, which means muscle fibrils, shows alternate dark and light bands. Yes, the next one is smooth muscles. Yes, in the right side we can observe the picture of smooth muscles. Yes, these muscles are involuntary, and we can observe the muscles are spindle shaped, and thread-like myofibrils arranged longitudinally. Thread-like myofibrils arranged longitudinally. Myofibrils, which means muscle fibrils, and sacrolemma with thin sacroplasma and presence of centrally placed nucleus. Sacrolemma with thin sacroplasma and presence of centrally placed nucleus, and the myofibrils lack of dark and light band. Lack, which means absence of dark and light band in these involuntary muscles. Yes, smooth muscles are involuntary muscles. These muscles are spindle shaped and thread-like myofibrils arranged longitudinally, and sacrolemma with the thin sacroplasma and the presence of centrally placed nucleus, and the myofibrils lack of dark and light bands, which means absence of dark and light bands. Yes, the next one is cardiac muscles. Yes, we know about the location of cardiac muscles. The cardiac muscles are cylindrical. Yes, in the right side we can observe the picture of cardiac muscles. The cardiac muscles are cylindrical and branched, and these are all involuntary muscles. So all involuntary muscles. And sacrolemma with thin sacroplasma, presence of centrally placed nucleus. As we can observe, the cell showing a sacrolemma, thin sacroplasma, and presence of nucleus in the center. Yes, and at the junction of cells, presence of intercalated disc, and number of myofibrils contain faint, dark and light bands. Yes, this is all about cardiac muscles. The cardiac muscles are cylindrical, branched, and these are all involuntary muscles, and sacrolemma are thin with the sacroplasma and presence of centrally placed nucleus. At the junction of two cells, presence of intercalated disc, and the myofibrils shows faint, dark and light bands. These are all about muscular tissues: skeletal muscles, smooth muscles and cardiac muscles. Yes, the next concept is herbarium. Yes, dear students, in first chapter, Living World, we studied about herbarium. So, herbarium is a dried plant specimen which is mounted on herbarium sheet. Yes, in the right side we can observe the herbarium sheet. So the herbarium sheet containing a dried plant. The dried plant is mounted on herbarium sheet. And for the preparation of herbarium, some steps are involved. Yes, preparation of herbarium involves steps. The first one is collection, pressing and drying, pressing and drying. The third one is mounting, and the fourth one is preservation, and the fifth one is labeling, and the last one is storing. Yes, these are all the steps involved in preparation of herbarium. Yes, the first one is collection. Yes, collection is the first step of process in preparation of herbarium. Yes, in this process we have to collect the plant. The plant should be has roots, leaves, flowers, and fruits. Yes, during collection process we can use some tools like a digger and a knife and scissor. See, these are all tools used to collect the plant specimen for herbarium. The second step is pressing and drying. Yes, during this process the collected plant specimen is spread out between the folds of old newspaper, and during spread out to avoid the overlapping of plant parts. After this process, the newspaper with the plant specimen should be placed in plant pressing for drying. After 24 to 48 hours, the presser is opened. See, these are all about the second step of herbarium process. The third step is mounting. Yes, during mounting process, the dried plant specimen is mounted on standard herbarium sheets, which means 41 to 29 cm. So, for mounting process, we can use gums or cello tape. Gums or cello tape. Yes, the dried plant specimen is mounted on herbarium, standard herbarium sheet 41 into 29 centimeter using gums or cello tape. These are all about a mounting process. The fourth one is preservation. See, after mounting, we have to preserve the herbarium sheets using 2% of mercury chloride. Using 2% of mercury chloride. And after preservation, we have to labeling. See, the labeling includes date of collection and name of the collector, and common name, and botanical name, and locality, family, and time, date and time collection. Okay, so the labeling process includes name of the collector and the date of the collection and time, and common name for plant specimen, and scientific name for, which means botanical name for plant specimen, and locality and family and time. See, these are all the steps including in labeling process. The final step is storage. Yes, after labeling, the labeled herbarium is stored in cupboards in proper position, according to well-known system of classification. These are all about storage. After labeled herbarium is stored in cupboards in proper position according to well-known system of classification. So these are all steps involved in preparation of herbarium. Yes, dear students, in this session we have discussed about diversity of animal tissues like squamous epithelial tissues and connective tissues, blood fluid connective tissues. It includes RBC, red blood cells and white blood cells types, granulocytes and agranulocytes, and muscular tissues include skeletal muscles, smooth muscles and cardiac muscles, and the last one is herbarium and preparation of herbarium steps, collection, pressing and drying, mounting, preservation, labeling and storing.